Okay, I'm going to answer those quiz questions on cells, and we have like 5 questions to answer here, and I prepared a couple of diagrams to use while I'm answering those questions. So, quiz number 1, compare and contrast apoptosis and cancer. How are they similar and how are they different? So apoptosis is a programmed cell death, a very rapid process to eliminate those dysfunctioning cells. And for example, when a tadpole changes into frog, and cells in the tail die through the apoptosis. And every normal cell goes through the apoptosis at some point, but cancer cells do not. So they somehow has this ability to resist or evade the signal, so they do not die and they just keep dividing and in a very uncontrollable way, and they just ignore those signals from other cells, so there can be a difference. And using number 2, using a hand-drawn diagram of the cell cycle, describe the major stages of the life of a cell and explain what happens during the S stage of the cell cycle. So this is the diagram I draw. So in the cell cycle has like 4 main phases, and the G1 and S and G2 is called, 3 of them is called interphase, and there is mitotic phase. So at the end of the cycle, this M cycle will be the cytokinesis. So it's the end of the cell division will be completed at that point. So G1 is mainly for the growth. So every cellular contents except the chromosomes will be duplicated at this point in this phase. And S is for the synthesis, DNA synthesis. So each of 46 chromosomes will be duplicated in this phase. This is a set of chromosomes and they will be duplicated, so like this. And G2 is there will be another growth phase, growth and little bit of preparation for mitotic phase. So they are just double checking for some errors and repair and getting ready for the mitosis. And mitosis is the cell division basically. So those duplicated chromosomes will be divided into 2 daughter cells. So at the end of the mitosis, there will be cytokinesis. It's a two complete daughter cells will be formed at the end of the mitotic phase, the cytokinesis. And yes, so number 3, what is the purpose of mitosis? Include a diagram of mitosis and explain what happens at each stage. So this is another diagram I draw. So in the phase there will be a chromosomes, the set of chromosomes here, and then the DNA will be copied into prophase, and chromosomes they just paired up, and then there will be a metaphase, and chromosomes will be lined up at the equator, and the spindle fiber from the edge will be connected. And there will be anaphase. So these sister chromosomes, sister chromatids, they will be pulled apart when you connect it to spindle fiber at the edge. So they will be just pulled apart, and there will be starting a little pinches here. So then there will be a telophase. Oh, this way here, the telophase. So the cell will pinches in the middle. Cell is just a introsome just pinches in the middle a little, and daughter cells will be formed. Each daughter cell, 2 daughter cells will be formed, and at the end of the mitosis, there will be a cytokinesis. So 2 identical cells will be formed, and this will be the end of the mitosis. Okay, so then, imagine number 4, case number 4. Imagine you are a medical scientist and are researching a cure for ALS. Explain how you may use stem cells to attempt to cure this disease. So this ALS is Lou Gehrig disease, also called Lou Gehrig disease, and that the disease affects nerve cells in the brain and the spinal cord, then weakened muscles, so they cannot really move the limb at the end later. As time goes by, and so the most likely application, stem cell application would be the transplant. So maybe using the stem cells to deliver growth factors, deliver those growth factors to motor neurons in the spinal cord. So transplant will be replacing those dying motor neurons of the patient with the new ones derived from the stem cell. So maybe the transplant will be working. And case number 5, describe an animal cell, including its major part, and ways in which materials are transported into and out of cell. So I just draw a quick diagrams here, a little drawing. So this is the animal cell, and there will be a nucleus in the center that controls the activities of the cell. So nucleus will be the center, like a city hall. They controls all the cell's activities, and there will be mitochondria, and which produce energy, and there will be cells is filled with the cytoplasm, and they are just the inner space of the cell is just it is called the cytoplasm, and the edge is the cell membrane that allows particles to move in and out, and there will be a ribosome that they produce proteins. They create new proteins, and this will be the lysosome, and that breaks down the worn out cells, so to use the cell component, and they reuse materials. So it's a kind of recycling plant. And this will be Golgi apparatus, Golgi complex. So it just Golgi complex, just they pass and transport to various parts of the cell, and then just to each part. And there will be endoplasmic reticulum. Endoplasmic reticulum is just that provides a network to move stuff to the cell membrane and back. And that's pretty much of all. And so there will be some ways of cell membrane transport, and the first one will be active transport. And there will be diffusion, osmosis, and filtration. and oh no, oh sorry, it was a passive transport, the diffusion, osmosis, and filtration, and there will be active transport, and they will use the cell energy, usually in the form of ATP, to move the stuffs in and out. And there will be the last one will be endocytosis, and exocytosis. So endocytosis is a small fragment of cell that they wraps around the particle to be ingested, and they just pinches in. And exocytosis will be the reverse. So there will be three ways of cell transplant.